Hello students, welcome back. We have completed viruses and we have left with viroids and lichens. So this is the last aspect of the chapter, biological classification. So in this class we are going to discuss viroids. You have already studied viruses, their properties, everything you have studied. Now we are discussing other acellular organisms. They are viroids. So what are viroids? Actually these viroids are the infectious agents. Like viruses, the viroids also, they are infectious agents. As they are acellular, we are not calling them as organisms, we are just calling them as infectious agents. And they have genetic material, but they have only RNA. They have only RNA as genetic material and they do not contain protein code. In viruses, you have studied that the viruses, they have enclosed by a protein coat called as capsid. But in viroids, we cannot find such type of protein coat. So here very importantly, the viroids, they are without protein coat. They contain only RNA without protein coat. This is very important, without protein coat. So we can define viroids like this. Viroids are the infectious agents which contain RNA without protein code. They are called as viroids. So what are viroids? They are infectious agents which contain RNA as genetic material but without protein code are called as viroids. And these viroids were first discovered by a scientist named as T.O. Diner. In the year 1971, T.O. Diner, he was the person who discovered viroids for the first time. Then what are the other characteristic features of viroids? Very interestingly, these viroids they are smaller than viruses. They are smaller than viruses. Actually, we are not knowing the size of the virus because I told you the viruses are smaller than bacteria, and you know the size of the bacteria. If the organisms, the viruses, they are smaller than bacteria, and these viruses are still smaller than the viruses. That is very important. The viruses are smaller than viruses, and they contain only about 250 to 370 nucleotides. So while studying the genetic material, DNA, RNA, you will come to know nucleotides. So actually this one you will study in the chapter biomolecules. There you will study what are nucleosides, what are nucleotides, everything you will study. So there you will come to know. You just understand here it contains only RNA and which is made up of 250 to 370 nucleotides. And its molecular, the genetic material molecular weight is very less. It is very less. So they are made up of only 250 to 370 nucleotides. And they usually cause diseases in plants. The viroids, they are only causing diseases to plants. We'll see what are the examples are the plant diseases which are caused by the viroids. So examples for the plant diseases caused by viroids are potato spindle tuber disease. One is potato spindle tuber disease. The other one is chrysanthemum stunt disease. And the other one is citrus exocarpus. So these are certain plant diseases which are caused by viruses. One is potato spindle tuber disease, chrysanthemum stunt disease, and citrus exocarpus. We are not going to discuss in detail about all these diseases. You can observe the images. So you can search in the Google or in the internet, so you will come to know how the disease will be observed. So here potato spindle tuber disease, the potato shape will be changing, it is like a spindle shape. And chrysanthemum stunt disease, it is seen in chrysanthemum plant, and citrus exocartes, it is seen in citrus plants. So when you look at the diagram or when you look at the image, you can able to understand so which type of disease is that and which is mainly caused by viroids. So it is one of the small topic that is viroids. If you get a question about what are viroids, viroids are the infectious agents which contain RNA without protein coat. And they were first discovered by T.O. Diner in the year 1971. And these are smaller than viruses. Viruses are smaller than bacteria, and these viroids are smaller than viruses, and they are made up of only 250 to 370 nucleotides. And one more thing, as they contain RNA, as they contain genetic material, they are capable of replication. So replication is nothing but duplication or multiplication. They can able to produce the same copy of that organism. So identical copies, that we will call it as replication or duplication. So these viroids, as they contain genetic material, they can able to replicate. They have the capacity to replicate themselves, and they usually cause diseases in plants, and certain examples are potato spindle tuber disease, chrysanthemum stunt disease, and citrus exocartes. So this completes about viroids. Now we will discuss the one more concept, that is the last concept in this chapter, that is lichens. Yeah, now we are discussing about lichens. Yeah, what are lichens? The lichens are the symbiotic association of an algae and a fungus. You studied the symbiotic association while explaining about the mode of nutrition in case of bacteria. So what is symbiosis? Symbiosis is nothing but the mutual benefit between two organisms. If there are two organisms, the one organism, it will be helping the other organism. At the same time, this organism is also helping the other one. So both are getting benefit. That is called as mutual benefit between two organisms is called as symbiosis. So such a association, it is the lichens. So the lichens are the symbiotic association. It is very, very important. The symbiotic association of an algae and a fungi. So lichens is equal to algae plus fungi, because the algae and fungi, they are mutually benefiting and they will form an organism called as lichens. That's why lichens are the symbiotic association of an algae and fungi. Here we should know, so how the algae is helping the fungi and how the fungi in turn helping the algae. That also we should know here. And in the lichens, the algal component is called as phycobiont. So whatever the algal component which is present in lichens, it is called as phycobiont, and the fungal component is called as mycobiont. So why they have given the names phyco and myco? Here actually, while studying about the algae, you will come to know phyco refers to algae. The study of algae is called as phycology. All these you will study in the kingdom Plantae. And myco refers to fungi. The study of fungi is called as mycology. That's why they have given these terms. Phyco refers to algae and myco refers to fungi. So this algal component is named as phycobiont and the fungal component is named as mycobiont. And one more thing, this is autotrophic and this one heterotrophic. Yeah, why it is autotrophic, why it is heterotrophic? Because the algae, this algae belongs to kingdom Plantae and And you know that all plants are autotrophic. They can able to synthesize their own food. Similarly, here also in these lichens, the algae can prepare the food, and that food can be provided to fungi. So how algae is helping the fungi? This algae, it contains photosynthetic chlorophyll pigment so that it can prepare its own food, and that food can be provided to fungi. And what the fungi will do? It cannot prepare its own food because it is heterotrophic and it is dependent on algae. Then how fungi is helping? So this fungi, it is heterotrophic in nature, and this fungi provides shelter for the algae. So fungi, it provides shelter for the algae. At the same time, fungi. In fungi, the entire organism is made up of thread-like structures called as mycelium. So with the help of mycelium, the fungi, it will absorb water and nutrients from the soil. So try to understand. So in order to prepare the food, it requires water and nutrients. So those water and nutrients will be absorbed by fungi and it is provided to algae. With the help of water, nutrients and sunlight, algae will prepare the food and that will be provided to fungi. So here both are getting benefit. This type of mutual association or this type of relationship is called as symbiosis. So the mutual association between two organisms is called as symbiosis. Here I think you can able to understand. Lichens are the symbiotic association of an algae and fungi. It is very important. You cannot simply write it as it is an association of algae or it is an association of fungi. They are the association of algae and fungi. The algal component is called as phycobiont and fungal component is called as mycobiont. So algae, it contains photosynthetic chlorophyll pigments so that it can prepare the food for fungi. And fungi, it provides a shelter to the algae. At the same time, it helps in absorption of water and nutrients from the soil. And this type of association is called as symbiosis. Then, where these lichens will be growing? So the lichens are usually growing on tree trunks, bare rocks, stones, etc. You might have seen in your villages or nearby. Usually on the trees you may have observed. When you look at the image, you will come to know, you may have seen the lichens. Here the lichens are classified into three types. So here we have three different types of lichens. So lichens are mainly of three types. The first type is called as crustose lichens. Second one, foliose lichens. Third one, fruticose lichens. So there are three types of lichens, namely crustose lichens, foliose lichens and fruticose lichens. So the example for crustose lichen is graphis, and example for foliose lichen is Parmelia, and example for fruticose lichen is Usnea. So these are the examples, but we should know where exactly these different types of lichens will be growing. So firstly, crustose lichens. So these crustose lichens, as the name indicates, they looks like a crust-like structure, and they are very compactly attached to the substratum or surface. They are generally growing on the rocks. The crustose lichens, they are generally growing on rocks and they are crust-like appearance. When you observe the structure, so here they look like this. Here the lobes are very small and it's very compactly attached to the surface. You cannot separate this crustose lichen from the surface. It is very compactly arranged. You can able to observe in the laboratory because these lichens you will study in your practical class also. So when you observe the different types of lichens, you can understand better. So crustose lichens are usually growing on the rocks and they are usually crust-like appearance and they are compactly attached to the surface. An example for crustose lichen is graphis. Then the second type, it is a foliose lichen. Here the foliose lichens are, they have leaf-like lobe structure. They have leaf-like lobe structure like this. They have leaf-like structures like this. So here the lobes are very deep. So these are the lobes. The lobes are very deep and these lobes are usually attached to the substrate. And these foliose lichens, sometimes they may be growing on rock. Usually they are growing on tree trunks and the branches. So when actually here the lobes are attached to the surface. It may be to the tree bark or the branches. So here these lobes are attached to the surface. And the example is Parmelia. So your foliose refers to, they look like a leaf-like structure. That is very important. They look like leaf-like structure. An example is Parmelia. And the third type is called as fruticose lichen. So here it is crust-like appearance. Here it is a leaf-like appearance. And fruticose lichen, it is shrubby in nature. So it looks like a small shrub. You may have seen, you know the difference between herbs, shrubs and tree. So this one, it is usually looks like it is a branch one. It is a branch one and it looks like a shrubby in appearance and it is usually hanging. So if one tree branch is there on the tree branch, so this will be usually hanging or pendulous in nature and they have branched structure. So this difference is very important. Here the lobes are very small, compactly attached to the surface. You cannot separate this one from the surface. And foliose lichen, they have leaf-like lobes, only lobes attached to the surface. And this one, fruticose lichen, they have shrubby appearance and usually hanging on the tree branches. Most of the time, the fruticose lichens are growing on the tree branches which are usually hanging or pendulous in nature. An example is Usnea. And one more important thing, these lichens, they have some fruiting bodies. They have some fruiting bodies. Here the fruiting bodies are the cup-shaped structures. So in all these types of lichens, you may have observed some cup-like structures like this. So here it can be clearly visible in the fruticose lichen. At the tip we can find, not only at the tip, here and there we will find some cup-like structures. These are the fruiting bodies called as apothecium. The cup-like structures are called as apothecium. So what is the importance of this apothecium? So these apothecium, they contain spores. When the spores falls on suitable substratum, it can able to develop into a new lichens. That is important. So like in fungus, like in mushroom, you have studied different types of spores. When spores falls on suitable substratum, it develops into new organism. Similarly, here in case of lichens, they have fruiting bodies, cup-shaped fruiting bodies called as apothecium, and they contain spores. When spores falls on suitable substratum, they can able to develop into a new organism. Then lastly, the economic importance of lichens is also very important. So what is the economic importance of lichens? Do you know these lichens are called as bioindicators? They are called as bioindicators. So lichens are a very good pollution indicators. These are the pollution indicators because they will grow only in the non-polluted areas and they will never grow in the polluted areas. See, for example, in one area, if you find the presence of lichens, you can say that it is a pollution-free environment because the lichens are only grows in the areas where there is no pollution. That's why if you find the presence of lichens, that area is usually pollution-free environment. If lichens are not growing means it is a polluted area. So in polluted area, the lichens will never grow. They will grow only in the non-polluted areas or pollution-free environment. That's why they are referred to as bioindicators. They are very good indicators of pollution because they will never grow in the polluted areas. Then other economic importance of lichens is, so these lichens are used as food, they are used as a fodder. So if they are used as food means it can be edible. Certain lichens can be used for edible purpose, become edible, and even it is used as fodder. Fodder in the sense it is used for cattle. So lichens can be consumed by us also, and even it can be used as a fodder for cattle. And other importance of lichens is that they are used as medicine, they are used in perfumes, and they are also used in flavoring agents. In certain masala spices, these lichens are also used. You may have seen at your home for preparing certain masala items. So these lichens will be used. Usually the folios lichens will be used. So these are certain economic importances of lichens. Let me discuss one more time. So here we have discussed viroids. You know, viroids are the infectious agents. They contain only RNA without a protein code, and they are discovered by T.O. Diner. They are smaller than viruses, and they are made up of only 250 to 370 nucleotides, and they are capable of replication. They causes only diseases in plants, and you know the plant diseases of plants caused by viroids. And later lichens. Lichens are the symbiotic association of an algae and a fungi. The algal component is called as a phycobion, and fungal component is called as mycobion. Algae prepares food for the fungi because it contains photosynthetic chlorophyll pigment, and fungi, it provides shelter to algae. At the same time helps in absorption of water and nutrients from the soil. This type of association is called as symbiosis, and there are three types: crustose, foliose, and fruticose. You know the examples, and you know where they are growing. The economic importance of the lichens is very, very important. They are called as bioindicators because they are growing only in the pollution free environment, and they will not grow in the polluted areas. They are used as food, fodder, also used in medicine, perfumes, and flavoring agents. So this completes the lichens, viroids and lichens. Now we will discuss few objective questions related to the today's class. See question number 110. Potato spindle tuber disease is caused by option A viruses, option B viroids, option C lichens, option D fungi. So here option B viroids is correct. Next question. Viroids is discovered by option A Ivanovsky, option B T.O. Diner, option C Bazarnik, option D Stanley. You know that T.O. Diner discovered viroids, so option B is correct. Next question. Viroids are option A free protein, free RNA, free DNA, free saccharides. So viroids are they have free RNA. They contain genetic material only RNA without a protein code. That's why they have free RNA. Next one. The molecular weight of RNA of viroids is option A high, option B low, option C very high, option D any of the above. You know molecular weight of the RNA is very low because they contain only 250 to 370 nucleotides. Option B is correct. Next question. Lichens are the symbiotic association between option A algae and bacteria, option B bacteria and fungi, option C algae and fungi, option D fungus and root of higher plant. So this is option C is correct, algae and fungus, because lichens are always association of algae and fungus. So this completes today's class, viroids and lichens, and so this completes the chapter biological classification. We have covered almost all concepts including the 5 kingdom classification. All the 5 kingdoms we have completely we have detailed studied we have completely studied all the 5 kingdoms. Later, the certain organisms which are not included in the 5 kingdoms are the viruses, viroids and lichens. So this completes biological classification. Thank you.